Polypeptide synthesis is the process in which peptides are produced to help create proteins that will assist in bodily functions. The most important aspect of polypeptide synthesis is the structure of the original DNA. This DNA is a key component to the process as it acts as a scaffolding for it to take place, determining what proteins will be produced afterwards. The structure of the DNA is a double helix. This is composed of two complementary strands of DNA to make up a chain of nucleotides. The nucleotides each consist of a sugar, a phosphate, and a nitrogenous base. The four different nitrogenous bases are adenine, cytosine, guanine, and thymine. These make complementary pairs with one another. They are bonded through hydrogen bonds, and A complements with T and C complements with G. Polypeptide synthesis occurs in two distinct steps, transcription and translation. Transcription takes place in the nucleus and begins when the enzyme helicase unwinds the double helix of DNA. This process breaks the hydrogen bonds linking the nucleotides together. After the deconstruction of bonds, the RNA polymerase runs across the template strand of DNA. When it's running across the template, the RNA polymerase is attaching loose RNA nucleotides of the complement to the template strand, creating a messenger RNA or mRNA strand. This strand is almost the same as the regular DNA, however thymine is swapped with uracil as the base. When the strand is made completely, it removes itself from the DNA and exits through the pore of the nucleus to enter the cytoplasm. Within the cytoplasm, translation begins with ribosomal RNA, also known as rRNA. Inside the ribosome binding to the mRNA, the rRNA and the mRNA then attract the transfer RNA, or tRNA, so they can match anticodons to codons. A codon is a group of three nucleotides that will code for a specific amino acid that corresponds with the codon. The anticodons are the complement of the codons and will attach to the matching codons. When anticodons find their respective codons, ribosomes catalyze the formation of a peptide bond and amino acids link. This process continues to occur until a complete chain is created. The chain will begin when the start codon and anticodon bond together. The chain will end when it reaches any of the three stop codons. This polypeptide will either fold to form protein or bond with other polypeptides and then folds to form a protein. Polypeptide synthesis ideally takes place in that process. However, there is a possibility for mutations to occur that will change what is produced. The more specific type of mutations are point mutations. Point mutations is when only one nucleotide is changed by substitution, deletion, or insertion in a sequence, which can result in a silent, missense, or nonsense mutation. Focusing on a missense mutation, which is when the point mutation causes one amino acid to be changed for another, usually occurring after a substitution. An example of a missense mutation having an effect on polypeptide synthesis is hemochromatosis. Hemochromatosis is the accumulation of iron in body tissues, which leads to fatigue, liver damage, arthritis, diabetes, and heart failure. The most common cause of hemochromatosis is a substitution on the HFE gene from C to T, changing the amino acid from cytosine to tyrosine, hence causing a missense mutation. Missense mutations usually make a protein non-functional or alter its function, and in this case, it stops iron absorption and iron regulation. This missense mutation shows that when polypeptide synthesis is completed incorrectly, it has a negative effect on the species and will have detrimental effects on the organism.